Hello and welcome everyone. Today we have really nice guest with us from Igem team Freiburg 2022, Jonas and Johanna, and they will be introducing about Igem Freiburg as well as introduce you about the program, what they are doing, and about their project. So let's get a start, and maybe we can first start with a brief introduction about you guys and also about your project. So, yes.
So, hello everyone. My name is Jonas, and I'm a bachelor student in Freiburg of biology, and I'm taking part in this year's Igem competition for the University of Freiburg, as well as Johanna.
So I'm doing my master's in biochemistry and biophysics. I switched from my bachelor's in physics to this master's program, and I chose to work with Igem to get some more lab experience and see how it actually works behind the scenes.
It's open to everyone at every stage.
We're a very mixed team, so you can have bachelor's and master's students. For Igem, you run in different tracks, so you'll have overgrad and undergrad, but that only goes by age. So if you're above the age of 23, you'll be the overgrad category. But we can take everyone. We have our bachelor students really impressed me this year. Like, they did so much and were so valuable for the team. So everyone, no matter of where you are in your bachelor's or master's, can participate a lot if you're just motivated.
Yeah, so all you need is curiosity and motivated enough to like pursue these projects, and main thing is asking questions. Why is it happening? And if you're curious, then for sure that's the thing. So that's the small introduction, but could you also introduce about Igem Freiburg team this year? How many team members are there in Igem?
Yeah, sure. So we are 11 people in this year's Igem competition for the University of Freiburg.
Okay.
And we're a mixed team, as Johanna already mentioned, of bachelor and master students. And I think, as you already mentioned, you should be highly motivated to participate, and curiosity is, I think, a key word which each of us has. So I think we are a small team compared to other teams, because some teams are even more than 20 people. So we are a small team, but I think we are all very motivated and very curious, and I think this is one key word which really describes our team very well.
That's nice.
We share similar backgrounds for this year's team. So we're mostly biologists and biochemistry, biophysics people. But we advertise everywhere. We even try to get some people from the technical faculty, maybe computer scientists, because there are so many different sub-jobs in our team that we don't even have to take scientists. Like, we could take people from business majors,
or any other field.
Everyone would be valuable, but this year we're pretty homogeneous.
Okay, so more like kind of interdisciplinary, like team members.
Yeah, so Igem is really interdisciplinary. So I think what really describes us is that you get a good insight what takes part of research in general. So not only the lab work, but also funding for your own research, the whole stuff about organizing your lab, and also what materials you need, but then also advertising the own project. We have to promote it in a video and also doing our own website. There are different parts which come together, and this is one reason why interdisciplinary teams can also be very good. So even if you do something which has no comparison with biology at first, you come maybe from graphical design or something like that, you can still participate in Igem. In the end, it should be that everyone of the team should have a brief overview of what we are really doing. So each of the team members should have an interest in biology, and in the end should
contribute a little bit in that project.
But it's really interdisciplinary and open to everyone.
That's really nice. I mean, people interested from, let's say, from other field other than biology, but they have a little, let's say, soft corner for biology. They can still participate in the Igem program.
So far we only talk about 11 team members. We also have 4 supervisors, so one professor.
They have both a doctor degree and work in the CIPS.
Okay.
So in the center here.
And one girl from the 2019 Igem team Freiburg, and she also helps us structure our project. And she told me a nice story about interdisciplinarity, because they had a computer scientist in 2019, and they even taught him how to clone. So everyone goes into the lab no matter your
Wow,
background.
that's really nice. Being a computer scientist and learning molecular cloning, wow, that's super nice. Wonderful. Yeah, maybe you can also talk about what are your goals, like future goals of your team and individual goals, let's say career goals.
I think for the team goals, our Igem project will be finished in the end of October with the giant jamboree where we present our work.
Okay.
After that, we don't really have further plans from Igem. It's offered that you can either go in the right direction of starting a startup or just further develop your project, but we don't have those plans yet because we are in the category Foundational Advanced.
Okay.
So we really need to wait what results we get before we can make other future plans. But there are some really nice and well-known startups coming from Igem, so they just build their idea further.
Yeah. Also the possibility to publish later on, but that all depends on how well is the project going. So at this stage we cannot say if our results we get in the end are really that impressive maybe or that good, so that we could go on with that. We already also spoke with one group leader of the university here, who is also interested in microcompartments, so it could also be a possibility to
later on
get a project going for further years, because it would be really nice. We are working the whole summer now on this project, and it would be really cool if it develops further. But we have to see.
Yeah, that's nice. It's kind of an initial stage what you are doing right now, and it can be on advanced stage and lead to publication, or as you mentioned, maybe that idea can be also used in some startups.
Yes.
That's wonderful. And what about your personal, let's say, career goal?
So as I'm in my bachelor now, I surely want to do a master's also in biology. My interests in the moment are synthetic biology, but also the cross field between biology and medicine. So especially working in the direction as you work, with signaling pathways and how they genetically influence, for example, or change in disease. And I want to further develop that. And my long-term goal would be to go into research, because especially in this project I discovered that I really love doing this, discovering new things. But that's long term. So first of all, I want to finish my bachelor, and then
it's always a step by step.
Yeah.
Once you, let's say, climb one step, then you think about there are many other things as well. I mean, there are many opportunities which you later on decide. But if you have a kind of rough goal, it's really nice, like doing masters or later on deciding and staying in research field. That's wonderful.
But I think especially when I mentioned it, the Igem project also showed me a very good impression of how research can actually be. So I think for each one who thinks about participating in Igem, it could be a good opportunity to discover if research is something you want to go in or not. Also maybe synthetic biology if it's interesting.
To add to that, it will not only show you the good sides but also the stressful parts. If you're struggling for months on end with one experiment that just doesn't want to work, so you see all of it and actually know if you can do a 5-year PhD program without getting into trouble.
It's like coins. We have two faces. One is like what we see, which looks very nice, working in lab and everything. Another part is frustration, stress, and other things. When you have deadlines and you have to work and you have failed experiments. But all is worth it at the end when you get nice results and when you are satisfied.
That's what I'm hoping to.
So all you need is that little bit of not little bit, you need more of self-motivation, and you should keep doing what you're doing, because that's the thing what you're interested in. And if you're doing that consistently, so consistency is much more important than even motivation. Sometimes
endurance and dealing with your failures in a way that doesn't make you feel bad but actually furthers your project.
You should accept the failure and you should learn from that, because it's always something you learn from your failures. So it's really important. And as I said, that motivation is not like a linear line. It always fluctuates. Sometimes you have very high motivation, sometimes it's down in negative. So what you should do is consistently work what you're interested in. Then for sure you will be successful and you will be happy and you can deal with all these failures as well. Say Igem program and how someone from, let's say, bachelor or even masters or any level of studies, their studies, how they can participate. So maybe we can just a little bit talk about a briefly introduction about Igem program, that what you need to do and how you guys decided to, let's say, participate in Igem program.
Yeah, so Igem is an abbreviation for International Genetically Engineered Machines, and it's mostly a competition. So the Igem competition was made to further synthetic biology and help young scientists and young researchers to find their passion, and also have this global get-together for everyone to share their experiences.
Yeah, and then another step which is made by Igem is it brings, so in the end, in this giant jamboree, which is a conference where all the competition participants come together, there are also different stakeholders from industry and research of the field of synthetic biology, so it also is a huge place where synthetic biologists can come together.
Okay.
And I think this is what Igem is about the competitional part, but also about this bringing synthetic biologists together.
And how did you guys decided? I mean, did you already had this team, or you guys knew each other from before starting Igem, or?
It was a bit different this year than other years because Freiburg used to have Igem teams consecutively each year, but then during Corona they stopped, so 2019 was the last team. And then for this year we just had two really motivated guys, one from biology, one from biochemistry, biophysics.
Okay.
We just went to the professor and said, we really want to do it, we really want to have a team, we know some guys that we know we can motivate to do it. And in the end they sent out emails and the professor agreed, okay, if you can get us a team, we will do it with you.
Yeah.
So we had those meetups and they were really hyping us up to do the project.
Okay.
So they called us and now we're there. I think for other universities it's just like an annual thing and it's already established, but this year we just had to push for it ourselves and just get into this whole thing all over again and restart.
And that's nice. I mean, those two guys managed to gather all these motivated candidates, and later on you will, hopefully you will find something interesting in this project. So how did you guys decide to do this Igem program? Like, did you get any suggestion? As you mentioned that because of these two members who are highly interested, but could you also maybe talk about, let's say, if anyone who's budding scientist and doing his or her bachelor's or master's, if they are interested in participating these kind of program, how they can participate? So is there any specific website or a specific requirement what they need to consider during the this team building and all these processes?
Igem also gives some resources and guides, so on the official Igem website you might find something that will help you, but maybe it doesn't fit that well for your situation. In the end, for us it was only through emails and knowing people that the information spread, and then you only needed to show up and have the motivation to be there. We didn't have any requirements for students except for them wanting to dedicate their whole summer to this project. But I think the more people want to join, the more limitations you will have. We were a small team and not that many people were interested to begin with, so we didn't have any requirements, but I guess if it was more people, there would have had to be some sort of choosing.
Yeah.
And it also, I think it depends mostly on the university you are at, how they sort their team out. So when it's established at your university already, then you can look out around end of the year before you want to do the program, because then most of the time they will be advertising the project and then you can apply at your university. When it's not established at your university already and you want to start Igem, then, as Johanna mentioned, there might be some informations on the Igem official webpage, and then you, like the two of our team, you have to find other students by yourself and also initiate something with professors, and it will be a little bit more work initially. But it can also work. So, for example, Tübingen, I think, they also initiated this year the first time, so it can work. And
be warned, Igem is not for free. There are also some fees you have to pay.
Yeah,
so
yeah,
you have
yeah,
registration fees and team member fees. So you will need to fund that somehow. Either university knows Igem and will give you funding for it, or you will call around and get lots of small sponsors, or do a mix like we're doing it this year. So there's also a financial aspect. And I've heard from some universities where the students will actually have to pay to participate. In Freiburg we were very lucky because university
gave you fund.
Yeah.
They gave us funding,
So the TIPS sponsored us with a huge amount, so we could start, and then we also have to find other sponsors because the research we're doing is expensive as usual.
It's always expensive.
But so in the end, as Johanna mentioned, we have to you have to find these sponsors. So most of the time, your own university hopefully helps you out. And there are also other things you have to keep in mind. So Igem really brings you many things, and you can learn very much of it. But you also you should dedicate your summer to it, especially when you are a small team, because then each of you have to do different things, and they all have to. You have to get them working before the end of the summer, because then the deadlines come. So you should really be motivated to spend a full summer into this project. So there are some things you have to keep in mind when you think about participating. But in the end, we think, I guess, that it's really worth it.
Yeah, indeed. I mean, it can be worth it because you have really nice team, and let's say further on, if you guys decide to do PhD or maybe stay in academia for research, then you will have already a nice team there, like not working together, but at least working in different cities or different country. Then you know that, okay, this person whom I worked in Igem, he or she is working in different area, and if I need to have collaboration, I can just call because I know that person personally. So it's really nice because in science, everything works with collaboration. If you want to do science alone, it's not that possible. So you have to have collaboration, because you can think in a different aspect, and other person can think or the point of view is different. So when you, let's say, work in a team, it's really like different brain answer one complicated question in very simple way.
That reminds me of our team structure. So you mentioned we're 11 people, but we are structured in 3 sub-teams who each work on their own topic.
Okay.
So, and we also have like meetings where each group tries to explain to the other groups what they're actually doing.
Okay.
I think that helps a lot because we're also then having experts and non-experts in our team to spark new ideas. I really like the structure we established there.
That's
and
really
I think it's also you learn many soft skills, how to work in a team and how to work with the others, and it also makes much more fun to work in a team. So
Indeed,
I think we have a very good team structure, and it makes much fun to work in such a team.
It's not only fun but also learning part as well. So you learn from each other. Let's say, as you mentioned, there are some people who are expert in that, and let's say if you're interested and not that much expert, you can learn, or you get, let's say, interested in other stuffs as well. So it's really nice opportunity.
That's the thing, especially for our team. No Igem experience will be the same. Every university handles it somehow differently, and we had very little guidance this year. So most of the lab work, especially in the beginning, it was just people from different fields saying, I used to do it like this in my lab, we did it like this, here I have a protocol for that. We're just learning from each other, and by now we're all on a good level.
Okay. So there was no as such like SOP or something, a standardized operating protocol or something
Not
that
at all. We
you
had
have.
to establish all of that ourselves.
Okay, that's really challenging. But it's really nice that you managed to do that. And it's a good experience. It's a good learning.
So at first it was very challenging. I remember one day when we 3 bachelor students had to do our first agarose gel, and it was like, hey, how should we do it, because we have never done it before. But in the end, when you figured it out by yourself, you can really learn much out of it. And if you come at other points later on where you have to figure something out because it's completely new, you are more confident with trying new things out, and I think that helps a lot.
And you make mistake, you learn from your mistake, and from my point of view, what I think is making mistake is good, but always make new mistakes, not same mistake. If you are making same mistake again and again, then it's not good. if you're making new mistakes every time, then you are learning new things every time, and then you will be like not expert, but at least you will know that what you're doing, where you did the mistake.
It's true. I'm most motivated on the days where we're not solving a problem but getting a new problem.
Yeah, this was the introduction about how to participate in Igem program and how you can participate, and what let's say a journey they went through and this Igem till this phase, and they are still in the middle of their Igem programs because it will be ending in October.
End
This
of
year.
October.
Yeah, end of October. So still there is time to do all this research work. But yeah, proceeding to next part, which is like talking about their project, what they are doing, but non-confidential one, because research is always kind of confidential. We don't want to share confidential information. But even if you want to know more about their project, you can visit their website as well as Instagram page, Igem 2022 Freiburg, right? That's yeah. I will tag as well.
And the website will be online towards the end of October.
Yeah, so it will be published at the end of October after project is finished. But before that, you can have a little bit of information about their project. So yeah, let's get started with the project part as well. So yeah, can you introduce us about your project, like kind of brief introduction?
Very brief introduction. So we want to enhance enzymatic production of certain chemicals, so enhance the pathways. And for that we chose a very recent discovery, those microcompartments, which are basically protein balls. And through they're just in the same space. We try to put our proteins inside of those microcompartments, which gives them a better spatial orientation towards each other, and they're closer together. So we're hoping this will increase the productivity of those pathways. And then to add on to this, as if this was not enough, we're also putting non-canonical amino acids into our compartments, and maybe even the enzymes. We haven't decided yet completely. And then we hope that they will even further improve
So improve the production, you mean?
Yeah.
He's in the pathways team. Maybe he can tell more about
So in the end, we want to enhance the yield and the efficiency of pathways in general. So we chose the indigo pathway to produce indigo, the blue dye we all have in our genes. Also indirubin, which is a derivat which also comes in this stage, which is studied as a cancer drug treatment. And also the trehalose pathway. So these are two pathways we are experimenting with. But in the end, our goal is to really establish these microcompartments that you can put your own pathway in it. And it gives you a higher efficiency of this pathway. And our long-term goal would be, or our vision is, that it can be an economical, friendly alternative to chemical synthesis. Because, for example, indigo today is mainly produced by chemical synthesis, and although that has many benefits, like it's cheap and you can do it in large scale, there are some downsides, severe downsides, like for example, it's all based on fossil fuels mainly, and also you have toxic side and waste products, and this all comes together and brings chemical synthesis in a not that environmental friendly illusion. And also when you think of that we have less and less fossil fuels, chemical synthesis can't be the future of how we proceed. So bioproduction of all these goods would be a really good
using microbes.
Yeah, would be a really good alternative, because for example for indigo we use L-tryptophan, an amino acid, as our substrate. So this would be very much beneficial in comparison to fossil fuels. And so our goal is in the end to show that we can improve the efficiency of pathways with these microcompartments and non-canonical amino acids, so that in the end
it will hopefully be competitive towards the chemical synthesis.
So that's really nice. I mean, that reminded me of one like old research as well. It's not research, it's like production of insulin. So previously, like people were using pig pancreas, and a lot. So that was not only like big consumption, but also it was affecting environment as well because of the waste product. But nowadays, these insulin production are mainly because of this modified microorganisms, bacteria. So which is like now more production, but with very less consumables, you can say.
Yeah.
Because I remember that people used to use like tons of pancreas from pig, and they were producing very little amount of insulin. But nowadays it's totally other way around. So you use very less amount of this, let's say, raw material, and you get nice amount of insulin, which is really nice, and that can be like helpful in your research as well, like what you're trying to build in micro compartments, like increasing the yield, which is nice. As you guys mentioned that you were also working on this unnatural amino acids, so could you briefly introduce, let's say, our non-scientific community who is, let's say, not aware of all the scientific terms, so could you like explain in simple words what are these unnatural amino acids?
So far we focus on modifying the microcompartments with an unnatural amino acids. So there are 20 canonical amino acids that will be usually used in protein production in your body. There are a bunch more of amino acids that are non-canonical, so not naturally occurring in your proteins, that have all kinds of different properties that can be exploited to further modify and improve structures. And we're trying to switch out a normal canonical amino acid with a non-canonical one to give our microcompartments novel properties that hopefully further improve them and help improving the yield even more. And we're doing it via the technique of Amber stop codon suppression. So there will be a sequence in your DNA, once it's read, that it will make the synthase stop, so the protein will be stopped being produced. This is the stop code. And we're just overwriting this stop signal with a signal for our non-canonical amino acid. So that when it goes on, we're just not taking the stop, but putting a new amino acid there.
Okay.
We're working on a
So
bunch of
that's
different
the
ones.
method for incorporating your amino acid into a protein.
Exactly, we're having a synthase specific for one of those non-canonical amino acids, and it overrides those stop codons because normal synthases, those are the ones that are carrying the amino acids, they won't recognize the stop codon.
Okay, and that will, let's say, improve, how to say, protein.
I can give you a few examples. We're working with different ones. So the one we're currently working on is called p-acetophenylalanin, and it is for click chemistry. So we just we mutated our DNA sequence for the microcompartment and mutated one place, so it will be the Amber stop codon. So instead of a normal phenylalanine, we'll put this with the click chemistry part, and then the click chemistry partner of this amino acid is a fluorescent dye. So you can easily label it without having like giant other GFP or other labels there, which is pretty neat.
Yeah, that's really nice.
And also there are a few other things we're exploring. There's also another one that's called BPA benzoyl. So this one will actually help stabilize our compartments. It forms under UV light, it forms covalent bonds to close by structures. We're not yet sure how well it will work and how well it will be incorporated at different positions. We will always need to be very careful to not destroy our protein by putting an amino acid at a place where it shouldn't be. So that's also where our modeling part comes into play. We're trying to kind of find out through simulations and modeling where it would be most viable or least detrimental to put a non-canonical amino acid.
Yeah, so as you mentioned that you can put this dyes or fluorescent dyes, so can we incorporate at like a specific position as well?
Yeah, exactly.
Like for example, for let's say if I want to study certain protein, so what we generally do is we tag a GFP with that protein, so we see expression pattern. So let's say in this case, because GFP is already a kind of big protein, so sometimes it can interfere with your specific protein function of that protein. So it happens if it is like tagged in a different, so either if you tag on C terminal, it can behave differently. If you are tagging on N terminal, it can behave differently. So that's why it makes complicated. So is it like kind of same with your, I mean, hypothetically?
Absolutely, that goes really nicely into what we've been doing our first early stage experiments. So we didn't start right away with microcompartments. We first worked with GFP and giving GFP non-canonical amino acids. And our plan was to try out different positions depending on where we will put it, that it will still fold if we put it in a place that's less conserved. So there will be parts in a protein that are not as structurally relevant, and through different generations and mutations, they will mutate a lot without killing the protein folding. So we're focusing on those areas to hopefully not destroy it.
Okay.
But for GFP, we've seen if we're going too close to the part that forms the chromophore, it might actually disturb fluorescence and then make GFP not fluoresce anymore.
Okay.
But we couldn't see that yet or prove it definitively. So we're still trying out. But of course, we're doing point mutations in very directed sites, and we're trying different positions because we are also hoping to see a difference if you put it at the start or at the beginning or somewhere in the middle.
Okay, well, that's really nice. Could you also tell about these unnatural amino acids as well? Like, are you also incorporating in these pathways what you're using?
So at the moment we are mainly focusing on the BMCs to influence them with the unnatural amino acids. We still want to try out in which patterns we maybe could also influence our pathway enzymes as well, unnatural amino acids. But as far as by now we are mainly focusing on the BMCs.
We're a bit hesitant to touch enzymes with non-canonical amino acids because their active center will be so specific to their function that might just really kill the function of the enzyme altogether. So he's working on a pathway group and they're now testing. We put a linker on our enzymes to actually put them into the microcompartment. And we're still needing to figure out if those linkers don't disturb the protein function. So we can't start putting all the things together there before we establish.
So one step by step.
One thing we actually thought of initially would be to, so at the moment we want to link our proteins into these microcompartments with a certain linker catcher, and we initially thought of maybe we could also do that via unnatural amino acid incorporation, click chemistry, but that's still an idea which might maybe come up later, but it could be too short our time because we have to finish that till October. To really incorporate that, so we are now focusing on using these catchers where we know of that they are really going into these compartments and really catching this the enzymes into the shell.
Okay.
So we are working with that.
Maybe to explain those catchers a bit more, our microcompartments, you can really imagine a bit like footballs because they're having those. They're different subunits, so they're like hexagons and pentagons forming this shell. And we're having a catcher on one of those pentagons, and this, while they're forming, they're also catching the enzymes and locking them inside of them.
So in general, you have this ball in the inside of different. So there are 3 different types of proteins forming this shell, and in the end, you have at one type of protein, you have always these tags. So we tag them, and then we have the catcher on our enzyme, and then when the structure is forming, then the enzymes will also be catched into the compartment. So then you have them in this shell, and then in close proximity to each other, enzymes, and this is what we hopefully get a yield increase because they are closer to each other, and thereby the substrates and intermediates are closer to each other and can be faster metabolized better.
Interesting. So how do you, let's say, monitor whether yield has increased? Or
this is a very interesting part because we chose our pathways also a little bit considering how good we can measure it, because there are a huge amount of pathways out there we could choose, but most of them require HPLC or mass spectrometry to really measure how much of that you have. But indigo is really cool there because it's a blue, you can see it and you can measure absorbance, and in the end we are doing calibration curves with the synthetic indigo to have a reference, and then we are measuring the absorbance of our at first we produce it in the cells, then we lysate them, and then we can measure the absorbance, and with our calibration curves we went the same way also with bacterial lysate so that we can compare it good, and then so in the end we can measure it with the absorbance. And for the trialose path there is a detection kit which also does some reactions so that in the end you have the isolated trialose, then you do this reaction, and then NADPH, NADPH is produced, and that you can also measure with absorbance. And so in the end we chose our pathways. On the one hand we want to choose something which is relevant industrially, but also on the other hand, how good can we measure it, because absorbance is something we could really measure a lot, and also good. We have plate readers over there. If we would use mass spectrometry as an Igem team, we can't go in a lab and say we want to do mass spec
it
every
can be a
week.
little bit expensive.
So, it would be expensive and also time consuming.
Indeed, and it's a lot of work. So it can only be done when you have more time. So you have limited time and, let's say, limited resources.
We were really happy to find the indigo pathway where, because it fits really well also with the measuring absorbance and all the enzymes and stuff. That is it's really lucky.
That's nice, wonderful project. So hopefully we will see some, let's say, improvement in this field, microcompartment field, bacterial microcompartment field, and maybe someday we'll see some publication or some maybe breakthrough from Igem team, or when you will go further for your research career. Who knows, maybe we will see something related to that. So you will see at least 2 members who are here, Jonas and Johanna. So they have already introduced everything about Igem program, about their project. But one last question which I would like to ask about, because in Igem program you have public engagement and science communication part as well. So talking about that, I would like to ask you what is your point of view on science communication, or how do you see as a science communication, or how you can improve, let's say, science communication. So could you just enlighten on that?
So a big part of our science communication project is under the motto kind of fighting fake science and research integrity, because given recent events with all the vaccine skepticism and what happened during Corona, as well as a very recent scandal in Alzheimer's research, where there were like manipulated images with thousands of citations and research money wasted in the billions. So we try to give different kinds of workshops and awareness for people on how to spot proper science, how to do proper science. Often research integrity and doing careful, properly planned experiments. It's not a thing. It's not you're doing it because you're a bad person. It's just you're doing it because you have not been properly trained or trained enough to do it in the right way. There's this problems of image beautification, which can be very tricky that people tend to do. Also quoting properly and just the training. And that's what we're trying to lean into and give awareness to the general public. Last week or 2 weeks ago, we hosted a workshop together with Embo, and she held like a 1-hour seminar for other Igem teams hosted by us about research integrity, what to look out for, and also positive examples. We really want to focus on, we don't want to spread mistrust for science, we just want to spread awareness on how to do it properly, and there are lots of positive examples where people do it beautifully. We're also going to schools and holding school lessons for like 10th, 12th graders, and trying to touch on this topic in a way that is understandable for them.
What's your point of view on science communication?
So I also really think that it's an important part, because we are, and that's also part of Igem. You have this human practices part, because I think important is that we are not only staying in our lab bubble, but also bringing the results we gain, and also the steps we get to our results, how we bring them to the public, to the general public, because in the end our results have no outcome if the general public doesn't accept them, and they also have to understand how research works. And I think we have all seen that during the COVID pandemic, that it's really important to understand how research works, how, for example, these vaccines are developed and that stuff. And that's also why in Igem it's part of the process. Not only do your lab work and modeling and all that stuff for that, but also the human practices part, that you get out of the lab and look, okay, how is our project, for example, reflecting the general public, but also, for example, like we approach with the research integrity topic we choose, to really also make a little impact, or at least induce some impact on topics which are related to science, but which are not really stay in the lab itself. And so I think we chose a beautiful topic there.
We got lucky in light of the recent events.
Because exactly the Alzheimer case now really showed that it's a problem which can, when it happens, can have a huge influence. It luckily happens in a little amount, so most of the research is done beautifully, but I think we still have to have this awareness, and especially in Igem, it's a good opportunity, because this workshop we did with other Igemers, and we are all budding scientists, so at this early stage it's, I think, the most important time to get this insights how proper research is done, because at later points it might be already too late. And then you have already done some failures which you will never can reverse the time, and you will have to live with them and
regret and all these things, and it's not a good practice. So as you mentioned, it's really nice that awareing people on early stages, which is wonderful, what you guys are doing, and you were like choosing this topic, and this news came of Alzheimer, which kind of correlates with your let's say focus on one part, and all about, like, I mean, at the end I would say Igem program is really nice. They're not doing only like science in lab, but also awareing people by science communication, and also doing this interactions with high school kids to aware more about science, and also research integrity and everything, which is wonderful. And I personally consider science communication is really a nice thing. We do, as a scientist, I mean, we do communicate our science when we publish our paper, but it's only to scientific community, because non-scientific community, they cannot understand what we have done. There is Western blot or qPCR or whatever. So this science communication part is really important, because there are many, like 80% people who are non-scientific people, but they do have importance in science as well. How they are providing fund, or they are supporting your science. So they should, it's our duty, being a scientist, it's our duty to provide that knowledge, but at least in simple words, that okay, what we are doing in lab. So previously scientists used to do that. So they used to like have a kind of conference or something, and they used to do small experiments and explaining that okay, what they are doing in lab and how it can help them further in future. And from that only they were raising funds. So that was the case previously, and still it's now. There are many people who are indirectly funding us, like paying taxes, and that tax, part of that tax is going into science and research area. So it's our duty to at least tell them that what we are doing in lab. We are not only just using pipette and doing these stuffs. We can show them, okay, we are doing basic research which can be helpful further, maybe not immediately after, like, let's say few months or so, but maybe a decade later it can be useful. Many research which are still important, and we consider that it's not that good or not that impactful. It can be helpful even in our research. It's not directly connected to humankind, but somehow further it can help for humankind as well, for further research or improvement of that research area. So yeah, that's all, and thank you for giving your time for interview session, and it will be quite helpful for people who want to participate and who want to know more about let's say your project and about science communication. So yeah, altogether it's really nice, and thank you for your time.
Thanks a lot for having us. And if anyone has questions still to us, you can hit us up on our Igem Instagram or even on Twitter, and we'll make sure to respond and answer to your questions.
Yeah, and don't forget to follow their Instagram page. Thank you.
Okay, thank you. Bye.